So I guess all the students hear me as well. Good morning, everybody. So today we will continue with developmental biology, signaling and development. So we'll start with a lecture of about an hour. Then we have, I think, 15-minute break, or let me have a brief look. 15-minute break. And then, as I said yesterday, I would like you to sort of turn on your camera if possible. Some people, oh, this is better, sorry. Do you still hear me, Shinya? Okay. 15-minute break, then you will have to look at slides that are uploaded on Adam in this picture folder. And I'll guide you through all of this. I look very much forward to this because I've done the pictures myself. Let's see how well that will work. So let's start with signaling and development. I'll go on this by first share screen. Here we go. And here. So, signaling and development, it's of course crucial that cells interact with each other, as you might see. It's crucial that humans interact with each other, which is not so easy these days. And I'm not sure we would like to further live in this world, but cells also have to interact. I think it's probably even more important for them because without it, really nothing would happen. And because this is so important, as a biologist, one has to know it. One has to know which signaling pathways, how do cells communicate. 5, 6, 7 languages, one has to know these languages. It's, of course, also important because cell communication means that molecules go from one cell to the other, which also means that in disease, you can sort of catch some of these important molecules at the outside of the cell, which is much easier with drugs and antibodies than with anything else. So it's not only important that cells know how to interpret signals and have ways to communicate, but these way of communication, because they happen by the outside of the cell, they're very important in medical aspects, because you can catch secreted molecules. You cannot so easily catch molecules that are made in cells, and we will talk about this extensively tomorrow. New ways maybe where one could do this. So we have to look at cell-cell interactions, how signaling pathways function at the molecular level, of course. What are the molecules? How do they interact? How do cells react differently to the same signal in distinct tissues? It's, as I said, there are maybe 7 different signaling pathways, and there are hundreds, if you consider the brain, thousands of different cell types. So 7 signaling pathways would not be enough if all cells would do the same thing once they see a signal. So each cell in its particular state will do something else with exactly the same signal, usually with exactly the same signaling pathway. And for many years, this was really puzzling. But now that has been kind of solved, how this works. What are the molecular consequences of signaling? If you consider a cell, which is sort of the basic unit of life, what can a signaling molecule do? Once it reaches the cell surface and maybe its receptors, it can change phosphorylation states, it can change localization, it can change localization in the cytoplasm, or it can change transcription. And when transcription changes, then the cell moves on its pathway and will not be the same as before. It will not necessarily return to the state as it was before. So transcriptional changes, they are usually relatively stable because you build up new proteins, and these new proteins have feedback loops in many cases. So these transcriptional changes, they really bring the cells from a state to another state. Cytoplasmic changes, they might allow a cell to move from A to B, but then once a cell is in B, it can revert to the state as before. So there are really conceptual differences between transcriptional changes and changes. Funny word, probably misspelled. And cytoplasmic changes. I think that's also good to know. There are different consequences of these things. Here, as I said, cell migration, mainly, or cell shape changes if we talk about the cytoplasm. Today, we only talk about transcriptional changes. And we'll look at these. Maybe on Thursday in the zebrafish, we talk about other things, more cytoplasmic changes. So here is an easy way of how a cell can become different during development. This is a way that does not rely on external signals. It's cytoplasmic determinants that are localized somewhere in the cell at the cortex, often at the cortex. Here it's just in the cytoplasm. And depending now of how the cell divides, if the cell division is in this direction, then of course these determinants will end up in one cell and will not be present in the other cell. And if these determinants influence transcription or influence cytoplasmic behavior, then these two cells are now different. This is a very important way for asymmetric cell divisions, for stem cells. Starts to be explored a lot in vertebrates as well, but was very well described first in C. elegans and Drosophila. The germline is usually determined like this. There are granules somewhere in the initial egg that have components that are needed for the germline, and they're segregated away from the soma. So the germline and the soma segregate away from each other because of such cytoplasmic determinants. So it's a very, if you want, the primordial way from a single cell to several cells and then the segregation to the germ cells. It's a very ancestral way, kind of an important way. But it's a rare way in many, many other developmental decisions. Because here you have to make sort of the decision, the cell makes the decision by itself. So this is important, but if we talk about cell communication, this is not a way where you make two different cells. This is such a cell segregation in C. elegans, how the P granules, which carry components which are important for the germlines, they're first, before fertilization, they're distributed throughout this egg, single cell. Here is the cell, the nucleus of the egg, and here is the nucleus of the sperm. The sperm just entered here, is now here just under the plasma membrane. And the P granules, they're still distributed equally in this one single cell. These two cells then meet, they migrate towards each other, they meet in the middle, and during the time where they meet and start to divide, the P granules are localized in one end of this still one cell. This is still one cell. Now this cell divides, the two nuclei are now here, and now you can see that the P granules are completely depleted of this anterior cell, and they're all localized in this posterior cell. Of course, the question then comes, how is that done? This goes via microtubular transport, actin-dependent transport, and this is a very important model system to analyze these asymmetric cell divisions in C. elegans. Later on, it always goes to a single cell. Here we have several cells, but again, these P granules are concentrated in only one cell, and this is the only cell that can give rise to the germline. At some point, of course, this germline cell is then fully determined, and then it can start to divide and make further germ cells. So it's a very nice way to do that, but in development, once you start to have 10, 50, 100, 1,000 cells, and each cell would have to have its own organizers, internal organizers for asymmetric cell division, that would maybe become too complicated. Anyhow, nature has used other means to have cells of similar fates, two A cells here, for example. Now, if only one of these two cells receives a signal, changes its transcription, becomes a B cell, and the other cell doesn't see that signal and stays an A cell, then this is, of course, a very elegant way how to do it. Then you can present the ligand to only a few cells from one side. Maybe here is a tissue that wants this cell to become an accessory cell. and then the proximity of the cell that secretes the signal and this responding cell makes this cell now an accessory cell maybe, and this cell here stays as it was. So these signals, that's what they generally do. They generate from equal cells, they select cells and tell them to go on in their pathway and do something else. Now these signals there are various types of signals. There are signals that go via diffusion or go via the extracellular space. They can go through the extracellular space from one cell to the other. They can also go much, much further than one cell. In some cases, they can go 10 cells, 50 cells. It's a bit controversial how far they go or how far they need to go, and we'll talk about this tomorrow. But so these molecules here, they're secreted through the secretory pathway, released into the environment, and then they interact with receptors on the other side, and then elicit the signal, and then these cells becomes a B cell and not an A cell. Now, of course, there's a lot of discussion of how freely molecules can diffuse. Tissues are complex. They are often folded tissues. So how is it that things can go along the fold? So there are a lot of questions, but this is sort of the textbook diffusion. So released from a cell, go into the interstitial space and disperse. Another way is direct contacts where the ligand, this signal here, the ligand is actually membrane bound. So it's not secreted by the cell, but it's put into the membrane, displayed then only on the cell that secretes it. So it cannot leave this cell, it cannot diffuse through the intracellular space. And if the neighboring cell has a receptor, then this receptor can interact with the ligand and signal. So this is proximity contact. Two neighboring cells can signal, and other cells from further away cannot get this signal. That's, of course, a nice way to have a signal that goes throughout the tissue and a signal that only influences neighboring cells. Now, there is a lot of controversy, a lot of interesting things happening here too with these signaling systems, because, of course, if a cell that is further away down here makes a philopodia, so a cellular extension, it can contact this cell from a distance. And as it appears, there are lots of signals that are actually bound to membranes, or they're lipidated and stay on membranes, and they might be contacted by cells that are a bit further away via philopodial contacts. So the classical way here is this is free diffusion, this is direct contact, but probably the better way is that this is somehow aided diffusion. Molecules bind to other things, they have been shed to not be degraded, so this is maybe facilitated or hindered diffusion. Direct contact, yes, direct contact, but the cell might make a philopodia and might be further away. But still, this ligand does not leave the membrane of the cell that secretes it. So there is, of course, a very clear distinction in, at least in the textbook version. Then there are other molecules that we hardly ever talk about, but that are incredibly important. Once a tissue is an epithelium, is this determined to be an epithelium, these cells have to communicate with each other too, apparently, and they do it via gap junctions. And I have to say, in flies, there's very little literature on gap junction cell-cell communication, but there is emerging evidence that this is a very important way to keep cells sort of along the same track. For if one cell is injured, maybe the next cell wants to know it so that it can prepare, or that it is sort of alerted, like with an infection. So then gap junctions couple cells to each other so that they kind of know what is going on. But as I say, in flies, we know very, very little about this. Very interesting, maybe something for you if you work on flies later on. So here we start. These are the molecules that are these molecules that are signals and tell. So you have a source cell that secretes the signal, and you have a receiving cell that responds to the signal. From my initial developmental biology lectures in the second year, you still know this slide here. So we have TGF-beta signaling molecules, large class. We have hedgehog, comes from Drosophila, of course, with this name. Jan Nüsslein called the mutant hedgehog. It turned out to be a ligand. It's not a secreted ligand, it's a membrane-bound ligand, or it's a ligand that has lipids attached to it, so it kind of stays in the membrane. So it's maybe of the second class direct contact, probably via philopodia, can go a bit further. So this is hedgehog signaling pathway. We will not talk about this. Wingless is another ligand that is lipidated, so maybe not very diffusible, not easily diffusible. So this is the wingless signaling pathway. Then we have Notch. Notch is the case. Notch in this case is the pathway has its name from the receptor and not from the ligand. These are the ligands here. These are the receptors. So the Notch pathway has its name because Notch was the first gene to be identified. Turned out to be a receptor, and the ligands were then delta and serrate, but the pathway got its name as Notch because of the first identified ligand. This is a pathway where the ligand is membrane bound, delta serrate. They're integrated into the membrane of secreting cells. They're recognized by Notch on the other cell, and very interestingly too, these two cells, they need to be also mechanically coupled. So if Notch sees its ligand and interacts with it, it starts to pull, and if the cell beside doesn't counterpull, Notch will not be activated. So there seems to be a mechanical coupling of after the interaction of ligand and receptor. So if you have secreted delta and serrate binding to Notch, although the ligand is bound to the receptor, the pathway is not activated because you cannot counterpull. A lot of interesting things that have been done to show that this is pulling and so on. So membrane bound, maybe lipid bound, and now here we go to this pathway. This is the one we'll discuss for the rest of the day and tomorrow. The TGF-beta signaling pathways. So the questions that we would like to look at today in the lecture, and mostly then also in the lab, and use a lot of P elements and transgenic flies. So I can repeat or reiterate or refresh what Martin said yesterday. The questions that we would like to address are: how are signals recognized? Who are the receptors? What kind of receptors are these? What kind of molecules are these? What kind of proteins? How is the signal transduced across the plasma membrane? This is, you can say it's a trivial question. It's of course an absolute key question in biology. Cells have this plasma membrane which helps them to be a cell, because otherwise everything would leak out. So this is a very, very strong boundary. And this boundary cannot be crossed by most of the molecules, or can only be crossed by few lipid-soluble molecules. You cannot bring a protein into a cell just by putting it onto the cell. There's no way. Pharmacy would like that, because then they could make antibodies or kind of binder proteins and inject it into the blood, and it would go into all cells. There is no way to bring proteins across a membrane, really, unless you have a receptor that binds to it and then sort of tries to bring it into the cell. But if it does it, then it's into an endosome, so it's also not in the cell. So the plasma membrane is an absolute border, and somehow if something on the cell surface interacts with an extracellular molecule, the signal, the interior of the cell has to realize that. So this is different in each of these pathways, signaling pathways, but we only talk about TGF-beta signaling. Then, of course, maybe the cytoplasm starts to realize that something has happened, but that is not enough for most pathways. The signal has to go into the nucleus. That's where the program is changed and the cell moves along. That becomes maybe more determined and eventually maybe differentiates, because it starts to express proteins that are important for this particular cell type. So the nucleus has, in most cases, will be affected. So the cell has to go to the cytoplasm and then into the nucleus. How is this done? Kind of interesting questions. There are not many things you can do with genes. You can either turn it on or you turn it off. You can turn it on a bit stronger, a bit weaker, but there is these two ways: turn off, turn on. very paradoxically, it turns out that, for example, wingless signaling cannot turn off genes directly. It can only turn genes on. So this is a bit funny, I find, because there are only two things you can do, and nature decided to not even do these two things. So how do you want to repress a gene with wingless? Then what you do, you activate the repressor. So you go through an intermediate step. But in the TGF-beta pathway, in flies, the same signal, the very same signal with the very same components can turn genes on and off, and we'll see how that goes. Is the cell inert without the signal? So, I mean, does a virus hit the population completely without protection? That's what seems to be happening at the moment. Cells seem to be very ready for signals, and they're very active even in the absence of signal. They constantly remember the cytoplasm that they have not seen a signal, so they're in the off state. That was not anticipated, because usually you think if nothing happens, nothing happens. But there is a lot of work to be done by the cells to remember them again and again that they have not seen a signal yet. And then we'll see how that goes as well. How is the cell sort of preparing to see the signal, and how is the cell keeping its state that it has not seen a signal yet? How do different signaling pathways interact? Well, this becomes complicated. We will not talk about it. How does the cytoplasm react to signaling? We'll also not talk about it in today and tomorrow. So now we start. This is the TGF-beta signaling pathway. That's what we want to look at now. One of these 7 pathways that we discussed today. All of these pathways have usually many ligands. So there most maybe in ancient times a single ligand in a very primitive organism. This organism has undergone genome duplication, so the ligand has duplicated as well. The receptor probably as well. So you have several receptors, several ligands. And in the TGF-beta superfamily, you have 35 members roughly in humans. So there's 35 molecules that look like TGF-beta molecules. And we'll discuss what they look like. So it's by their structural fold. And of course, this in turn a bit translates into amino acids, or the amino acids into the thing. So this is, I don't know, you can see that. This is, I'll come back to this in a minute, but if a molecule looks like this, here, this is one part of it, and this is the other part of it. If you would crystallize thousands of molecules and you see one that looks like this, and it really looks, it's 2 molecules that bind like this, then it's a TGF-beta molecule. That's their characteristics. The 3-dimensional fold that they have makes these molecules TGF-beta molecules. And there are 35 genes in the humans that encode molecules that look like this. This is one single monomer encoded by the gene, and then this protein during the secretory pathway dimerizes and then becomes like this. So all molecules that look like this, there are 35 different genes encoding molecules like this in humans. There are 7 of them in flies. And they're not only pink molecules like this, but they're also blue molecules look like this. So they might have a few amino acids that are different here in a few of the ligands and different from some of the other 35. So the families that look like this are usually subdivided into subfamilies that look even a bit more alike. So in the TGF-beta family, there's a BMP, bone morphogenetic proteins, a BMP family. There is an activin family. There is a TGF-beta family in the TGF-beta pathway. In flies, we have only 7 ligands. There are here DPP. This is the Drosophila BMP homolog. There is another one called 60A here. It's also looks like a BMP. It has homologs in mouse and human. So there are 2 BMP type molecules, actually 3. There's another one here, screw, I'm sorry. There are 3 BMP type molecules in Drosophila. We will only deal with DPP, the DPP molecule, or the DPP gene, which codes for one of these which codes for the monomer. This monomer then dimerizes and forms functional DBP molecules. As it turns out, DBP often heterodimerizes with GBB. GBB is, where's GBB? It's not on this slide. So let's forget about it. It doesn't really matter. We will talk about this molecule here, DBP. So you can see again, when you go and compare all the BMP molecules in, let's say, invertebrates and vertebrates, you can see that DBP is very similar to human BMP2 and BMP4, and they can actually be substituted. If you inject BMP2, human BMP2, into DBP, into flies, then it will act like DBP. If you synthesize DBP, we've done that many years ago. Also, you make the protein and the dimer and you purify it, then you can make bone in human cells, because that's what BMP2 does. So these are interchangeable. Quite surprising, meaning that they have the same receptors as well. They recognize the same. The Drosophila protein recognizes the human receptor, and the human ligand recognizes the Drosophila receptors. So when I started in my lab 30 years ago, maybe, DBP was well known as a gene, but there were no receptors. Nothing was known about signaling in general. It wasn't even known that hedgehog is a secreted, also is a ligand. So it was the beginning of everything. DBP was known, there were no receptors known, there was no signaling pathway known, no nuclear components. It was just a black hole. And what we do in the course today is to see a bit how this black hole was sort of using genetics, how we could decipher this whole logic of this DBP pathway. First, who are the ligands? Who are the receptors? Who are the mediators? How do they regulate genes? We were the first ones to try to figure out how genes can be regulated by DBP signaling. And that's what we do then in the course afterwards. So this is the pathway, the BMP signaling pathway, or the DBP pathway for us. Here are BMP ligands, here are activin ligands. All of them belong to this TGF-beta pathway, and they use very similar components. We deal with the BMP ligands here, over here. They're either homodimers or heterodimers, but always dimers. The monomer does not interact with the receptor. I think the monomer is also not secreted, so monomers are not viable proteins. They're probably too sticky as well, so they're always dimers. Often heterodimers, probably often homodimers as well. This has not been completely sorted out. That's very difficult to address. Who is a monomer, heteromer or homodimer or heterodimer? Anyhow, they're dimers. These dimers bring two types of receptors together. So with these dimers, they swim around the cell, and if they are recognized, if a cell has receptors for TGF-beta ligands or for BMP ligands here, then the ligand will bring two types of receptor in close proximity, a type 1 and the type 2 receptors. Now, for many signaling systems, the receptors are exquisitely expressed in a few cells, also in specific cells. So only some cells can respond to some of the ligands. Generally, in the BMP or TGF-beta signaling pathways, most cells express receptors. So the cells are kind of ready to see a ligand. These ligands are then secreted very locally and affect neighboring cells. But almost all cells can respond to DBP, for example. All cells in Drosophila can respond to DBP, most of them, I would say, because they have the receptors. If they wouldn't have receptors, they can't respond. But most cells express type 1 and type 2 receptors. And then when they see a ligand, these two receptors come together on the cells that are surrounded by ligands. These two receptors cross-phosphorylate each other. We'll discuss all of this in quite some detail, how that goes, because these are schematic schemes, but there are molecular logics behind. I mean, why would this recognize this? Why would this transphosphorylate this? It's all about amino acids. It's all about structures of proteins. And we'll have a look at that. Then the cell has realized that there is a ligand outside because it clusters receptors. these receptors transphosphorylate. They also phosphorylate a molecule that is also present in most of the cells, which is a SMAD protein. This is a cytoplasmic protein that is ready to become phosphorylated upon interaction and activation of these receptors here. This molecule becomes phosphorylated, probably as it interacts with another molecule called a SMAD. These are SMADs. MAD was the first Drosophila mediator that was identified, so it was called MAD, Mother Against DBP. It was a genetic coining again, because DBP was a certain mutant, and they found a mutant that suppresses it, and so it was Mothers Against DBP. Small was a gene from C. elegans that turned out to be similar to MAD once the things were sequenced. So the two SMAD, small, and MAD were combined to come up with the name SMADs. Small were mediators of TGF-beta signaling in C. elegans. MAD was the mediator in flies, and that resulted in the name SMAD. And it turned out that there are co-SMADs that are not being phosphorylated, but they almost look the same. They make probably hexamers, trimers or hexamers, but multimers. So maybe trimers, hexamers, which go to the nucleus, bind DNA, and regulate genes. So it's a relatively simple pathway: ligand, receptor, mediator. You always need a signal. You always need a receptor. You always need a mediator. The easiest way would be to have the ligand bind to a receptor, and the receptor is cleaved off and itself is the mediator and goes into the nucleus. That would be the simplest way. And that's what Notch does. Notch interacts with its receptor, is cleaved here, goes into the nucleus, binds to DNA, and regulates genes. Here, it's the second simplest way. You have one of each: a ligand, a receptor, and a mediator. DBP, DBP receptors, SMAD, and then you're in the nucleus. So it's relatively simple. But what we would like to do now is go into this pathway, look a bit at the molecules. What is their properties? What are their structures? And then elaborate on it a bit. So again, TGF-beta signaling components. As I said, when we started, there was DBP was known as a gene, and that was it. Nothing else. And then slowly, people started to clone genes and find similar genes, and then something that looks similar to DBP is probably a ligand. The sequence is similar. So these are the three ligands of the BMP pathway in flies: DBP, GBP, screw. There is an activin pathway, which then was called deactivin, Drosophila activin, because it was found after the activins were found in vertebrates. So this name was then co-adopted from the vertebrates, while often it is the other way around. But here it was cloned after the genes in mammals were characterized. And then not characterized is probably not true anymore. In the meantime, Maverick, Alp, what is Alp? Is Shinya still there? Anyhow, there are three genes that have not been characterized all that much. We talk about this one here. The receptors we talk about are thickwin and punt. As you can see, there are other receptors, but for DBP, the most important receptors are thickwin and punt. Then we have MAD, which is the mediator, which associates with the co-SMAD, which is Medea. So we'll talk about DBP, ligand, thickwin, punt, the receptors, MAD, and Medea, the mediators. And of course, if you look at something like this here, it's every cell that sees DBP activates thickwin, punt, co-SMAD and MAD go into the nucleus, and then you say, okay, every cell regulates the same genes. If these proteins bind to DNA, then every cell that sees a DBP signaling molecule does exactly the same thing. Could have been that way. Often cells do regulate the same genes. Often they upregulate their own receptors to make the cells more sensitive, and all cells upregulate the receptor for various reasons. But if you want to make cells different and every cell responds the same way, you kind of have a problem. And if a cell that is determined to become a neuron responds to DPP the same way as a cell that is an epithelial cell, that also wouldn't make sense. These are two completely different cells. But for many, many years, the question was, how does the same molecule, SMAD, co-SMAD, this is one molecule, this is a trimer or a hexamer, why does it do different things in different cell types, the same molecule? And we'll discover that during the course today. And then in the afternoon, we sort of make it clear how this whole system works. So again, DBP, Thicven, and PANT as the receptors. Mothers against DBP is the mediator, MAD, which is phosphorylated by these receptors, goes with Medea into the nucleus and regulates tissue-specific target genes. So different genes in the gut. We'll talk about gut genes in the morning. Tomorrow we'll talk about wing-specific genes, or also this afternoon. So a mid-gut cell in the Drosophila embryo will respond differently to DPP signaling than a wing blade cell, then a nervous system cell, then a germ cell, then anything. All cells respond differently to these very, very same molecules here, to these MAD Medea eventually that go into the nucleus. And we'll find out how that works. So now we start with the ligands. What are the ligands? We go briefly through the ligands, the receptors, and the SMADs, and then we are ready for the course. Let me try something very sophisticated. What happened now? Oh, you see this? What I wanted to do is to use my whiteboard. How do I do that? Stop share. Here we go. Okay. First try. This is my fantastic whiteboard. So now we talk about the ligands. We talk about these molecules here. This is a dimer. A dimer is not encoded by a gene. A dimer is a protein consisting of two single molecules. So the gene, DPP, encodes this molecule. Probably must encode this entire molecule, but it might actually encode a much longer molecule that is then cleaved and results in this molecule here. And this is actually the case. So here we have, let's say, sorry. This is the DNA, it's a double helix. Here is the DPP gene. I'll use more modern technology this afternoon. Should be italics because it's a gene. The protein would be written like this with DPP. So proteins are usually capital letters. Genes are italics and small letters. So if this is the DPP gene will of course have a transcription start site where transcription starts. Then it will have a few introns and stuff. And at the end, you would like to get this protein here. DPP is a ligand. So if DPP is a ligand, it has to be secreted. If we would be in an online course, not in an online course, I would look into the audience now and pick a person and would probably ask him how do you make a protein secreted? I mean, if every protein would be secreted, the cell would be empty. There would be nothing in there. But of course, only very few molecules are secreted. And DPP, the protein itself, is 588 amino acids. 588 amino acids. So, I mean, you go to the gene, you look at the sequences here of the introns. You probably find out on flybase what are introns and exons. Then you translate the mRNA without the introns, so the major mRNA. And then you find, okay, there is an open reading frame from 1 all along without stop codon to 588. And then you have to admit that, yes, this is the protein that is probably made. knowing that DPP itself is only 112 amino acids. So the ligand is only 112 amino acids. So some things must happen to that ligand. Obviously, a secreted molecule has to have a peptide, a signal peptide, which brings it across the ER. Translation happens at the ER for these molecules. The molecule has to go, the protein, as soon as it's translated, has to go to the ER. And in order to do that, you have a signal peptide that is translated in the beginning. You see, a signal peptide. Now, these signal peptides are usually lipophilic or hydrophobic, usually 20 amino acid length long. And if you just go through the genome and find a translated sequence, and you find hydrophobic amino acids, 20 of them at the N terminus, you will probably say this encoded protein has a signal peptide. And DBP needs a signal peptide because it has to go into the ER. These 112 amino acids here, they're at the very end of the protein. So the last part of the protein is actually what is the mature ligand, 112 amino acids. So there is a whole large domain here that apparently is cleaved off in the ER, presumably. So here there are several cleavage sites that can cleave this protein in the ER, and furins do that job, which separates then this molecule here from this one and sort of allows to release the mature signaling protein. Why would you do that? Why would you make a 588 amino acid protein to discard 1/5 of it and throw it away? This is energy-wise not very smart. As it turned out, if you make a mini gene that only has a signal peptide and this part here, you can do that in flies. See what happens? Nothing happens. There's no signaling molecule that is secreted. So apparently this region is important to generate the mature ligand. And as it turned out, this pro domain here, the pro domain of DBP, which is longer than its ligand, is actually domain. This pro domain is required for DBP to fold properly. And DBP has a very, very funny fold. It's a cysteine knot protein. And I probably, we'll see it afterwards. So I have to go back now. Stop share screen. Stop share screen. Here we are again, and now we share this screen again. Okay. So what I told you is that DBP, it's a gene. It first is translated as an mRNA molecule. This mRNA molecule is spliced. It goes to the cytoplasm. The protein that is translated at the ER is a large protein. It's a pre-pro protein. It dimerizes before secretion. Mostly heterodimers of DBP. There's also some homodimers, also some heterodimers. The signal peptide, the first 20 amino acids, are required for secretion. Then it's good. You go into the ER, you're in the ER. The pro domain, signal peptide is cleaved off. The pro domain is required for the folding of this C-terminal future mature domain, and is then cleaved off once the molecule has formed, and maybe or presumably before the dimer folds. I think that's not entirely clear. It's also not even clear in flies whether the pro domain remains actually associated with the cleaved domain. The pro domain, helps the ligand, this ligand here, to fold. And for many TGF-beta molecules, it stays associated then. So it allows this to fold, it allows this to dimerize, like this. And then it remains associated because it's actually a ligand of the ligand. If this is DBP, the pro domain which helps it to fold is sort of a ligand. It's also often secreted in TGF-beta. It's always associated with it, unless in the plasma of our blood, TGF-beta needs to be activated, and then the prodomain leaves and the ligand is freed. In Drosophila, we don't know yet that much about it. So in the course, it's a cysteine knot molecule. It's difficult to explain how that is. We'll have a look at it. So DPP is this ligand. It has several antagonists are known for BMP ligands. You know best noggin, cordin, very important for dorsoventral patterning in vertebrates. And Henry will talk about this in fish. So of course, this ligand, it binds the receptor. But if nature wants this ligand to be made, but not immediately recognized, it makes antagonists that bind at the same place where the ligand would bind. And then the ligand is shed, it cannot bind. You first have to take off these antagonists to free the ligand and bind to the receptor. In fish, the antagonists are made only in one side of the embryo and not in the other side. So the molecule is shed, cannot bind to the receptor on one side of the fish early embryo, but it can bind on the other side. So there are various ways you can regulate the activity of these ligands. The best way is antagonists. Antagonists block receptor activation because they sort of masks the sites where the receptor would bind. This is what DPP looks like. I mean, you've seen it here now. So this is, yeah, it would be like this. So the orange part here is one of these molecules. So this is the orange part, like this. This orange part here is this molecule I have in my hands. And then the blue one is the one that dimerizes with it. So you have this part is the orange, this would be the blue, and they're held together by a cysteine bridge. And this cysteine bridge is green here in the thing. So they're the two molecules, they fold in a way that they sort of snuggle into each other. But in order to be sure that they don't dissociate anymore, there is a cysteine bridge that is made between these two monomers. And then you cannot put them apart anymore. They're linked, they're physically linked. The two chains are physically linked. And this is this green link here. This is a stereo picture. And in last year, of course, we had stereo glasses, so you could look at them and then you would see this in 3D. Yeah, I cannot send you, I could have sent you all a stereo glass by mail, but maybe in future days, every student will have 3D goggles anyhow to go for virtual reality, and then you can go by virtual reality into these two things. I love it to look at three-dimensional molecules with these glasses, because it really tells you that why DPP is because it falls like this, because it has each and every amino acid here, makes it a bit different from the other 7 ligands. They have the same fold, they look all like this, but it's the amino acids are a bit different, and then they might bind a bit different receptors. So it's all in the three-dimensional structure, it's all in the amino acids. So structure, I love structural biology. It's all into three-dimensional structures. Of course, a lot of things are not structured in structure, and they might be the most important things because they structure upon recognition. So these induced fit. But I asked once a very famous German scientist what he thinks is sort of the essential of life, and what he told me is that it's all in these three-dimensional structures and in the way two molecules would interact under certain conditions, maybe at a certain level. It's all about intramolecular recognition or not. And this is the protein world. It's probably true for the DNA world. And that's why I like structural biology, because there you can see these interactions. Changing one amino acid at a very critical site, changing this cysteine here to not make this molecule completely linked, and you're dead. And there is a structural logic to it. There is a structural logic to every intramolecular interaction, and all cells do is to regulate highly in a regulated manner. They regulate protein-protein, protein-DNA interactions, intramolecular interactions. And you can only understand these by structural biology. So I'm absolute fan of structural biology, but I'm a fan of developmental biology because these structures only make sense once you look at them in a multicellular animal or in development. So developmental biology is sort of understanding the logic of structures in development, in real life, in real animals. So I think, yeah, whenever you see a structure, have a look at it. It's funny enough. It's all the logics is in there. So that's what DBP looks like. And the logic is in there. It has certain amino acids and they will recognize the receptors. Otherwise, there would be no interaction. If it wouldn't be folded like this, it wouldn't work. This we can maybe skip. Are again these folds. So this is BMP2, BMP7 in red, BMP2 in blue, TGF beta 3 and TGF beta 2 in 2 other colors. And when you overlay the backbone of these molecules, you can see why they are called TGF beta molecules. They have the same fold. They don't have the same biological activity because they have different amino acids exposed on these folds. And that means they can recognize some receptors and not others. But if you have a molecule that has this structural fold, it can be nothing else than a TGF-beta molecule. Might be even an agonist that dimerizes and inactivates these heterodimer, whatever. But it's part of the class of TGF-beta molecules. No doubt. That's the definition of families of proteins. They have the same fold. Interestingly enough, so this is kind of a funny drawing of, this is the dimer of BMP7 here. Yeah, it's a bit like this. You might not recognize it so easily here. And the first postdoc I ever had in my lab, he purified DBP in the lab. It was very difficult, and Roche only had, no, it was Novartis or Ciba, no, Novartis, had a protocol to purify TGF-beta. TGF-beta 1. TGF-beta 1 is important for human health, so they made a lot of efforts to purify this. It's of course not easy because you have a protein that has a predomain, a prodomain, a single peptide, and it falls in a very complicated manner. So you cannot just put it into E. coli and it will fold. So they developed a protein to express only the ligand and then shake it in cold water until it finds its proper sort of way to dimerize with the cysteine bridge. And that would result in a very fine band. And this fine band, they showed, is the mature dimer. So they elaborated protocols to purify the dimer. And we did that, Jay Groppe in the lab did that with DBP. Then he left the lab and went to Stanford, to the Scripps in California. And what he wanted to do there is sort of become a structural biologist. Also here, I couldn't make anything else with him than a structural biologist. He was always in the other labs. He didn't want to do developmental biology. And then he went to the Scripps. And since he knew DBP very well and wanted to show that DBP is active, he used noggin, an antagonist of BMP2, to show that DBP binds to noggin, arguing that if DBP can bind to noggin, then it's active, it's properly folded. And then he was supposed to establish the structure of BMP7 at the Scripps, and it didn't work. He couldn't get this very active, and then he said, maybe I can purify this few molecules that are active by taking them and letting them bind to noggin. And he used this trick to purify BMP7, but then what he did is to solve the structure of the noggin BMP complex, because that's the only way he could have his BMP7 active. And then he got two things. He got the structure of the BMP7 here, and he also got the structure of noggin. And as you can see, noggin binds to both monomers, binds to a lot of sequences here, which usually would be bound by the receptor. So when noggin is bound to DBP, these surfaces here, this comes from the back to the front. The receptor would interact with BMP7 here. It would interact here and on the other side, but all of these sequences are blocked by noggin. So nicely showing how this sort of Schmetterling, he called it, structure of noggin can inactivate the ligand. Again, it's all about molecular interaction. As it turned out, DBP, all TGF-betas are cysteine knot molecules, because also the monomer itself forms cysteine bridges in a very particular way. It turned out that noggin also is a cysteine knot molecule. So there are a lot of evolutionary speculations that the ligand and its antagonist evolved out of the same protein family. Kind of intriguing that ligands and their own antagonists through evolution come from the same initial gene. Will be kept in mind when we try to see what very ancient organisms look like. So not much has been done with this theory, but it was a very interesting, it was a Nature full article, was a very interesting paper because it showed this structure plus this, plus the whole logic of noggin being an antagonist, and again, structure explaining biology. Here again, some of these figures, but since this again would be a 3D model with these glasses, but you can maybe go to the internet if you want. Maybe I'll do that during lunch break and show it to you later on. That's about DBP. So all we have to explain now is how a molecule of that structure here can elicit responses, signals in receiving cells. For this, we need receptors, and these receptors we have seen, they're called pumped in Drosophila and thick veins. So a little bit of the history. When I started my lab, as I said, DPP was known. It was known to be a gene that generates 15 phenotypes, deca-pentaplegic. That was the name of the gene. The name came because all the imaginal discs, and Drosophila has 15 imaginal discs, 2 for eye, 2 for wing, 2 for, you can calculate it's 15 imaginal discs because there is only one genital disc. So it's not a paired number. All these imaginal discs were disrupted in the absence of this gene, DBP. So it was given the name deca-pentaplegic. All 15 imaginal discs are defective. It turned out when DBP was sequenced that it had an amino acid sequence that was very similar to TGF-beta 1, the first ever cloned ligand of this whole family. So people speculated that the DBP gene, which affects all imaginal discs, actually encodes for a ligand. That's when I started my own lab. There were no receptors. There was just a proposal that DBP is a TGF-beta ligand. So 3 weeks after I started my own lab, what I wanted to do is to figure out how DBP signals to cells. So there was a DBP mutant. There are some things that were not good in this embryo, and maybe one could screen mutant collections to see other embryos that also have the same defects, and maybe then the receptor would be among these. That was the proposal. So I went to Zurich, gave a talk, and in the audience was a guy called Konrad Basler, who also had just started his own lab. And I proposed that maybe by looking at embryos and a lot of available mutants from the Janni Nüssleins collection, maybe we could figure out who the receptors are by similarity. And then after the lecture, Conny Basler, he came to see me and said, as it appears, we seem to have the same aim. I also would like to figure out how DBP, this putative ligand, signals to cells and how it affects imaginal disc development and so on. And he said, actually, what I have in my fridge is clones, PCR amplified clones that contain sequences that look similar to the first TGF-beta receptor cloned in mammals, TGF-beta receptor 1. So he said, I have 5 tubes with each of them has a clone of a Drosophila gene that has similarity to this TGF-beta receptor. And maybe one of them is the DBP receptors, we don't know. As it turned out, one of the clones was pumped. The other clones was thick vein. The other clones was yet another DBP receptor. So he had all the DBP receptors. He had all the receptors in there. What I did, I asked Jannie Nusslein to send me all her collection of mutants, 150 mutants she had, wingless, hedgehog. I mean, these were just names. Nobody knew what it is. There was a mutant called Pond. There was a mutant called Thickvein. And sure enough, we didn't know. We looked at these 150 fly stocks, collected embryos from heterozygous animals. 1/4 of these embryos were mutants. We didn't know which ones they were. There were no balancers that had markers on them in those days. But we just looked whether a quarter of the embryos had phenotype similar to DPP. And we found some. And one of these was Pond. As it turned out, that was the gene that Connie had the cDNA for it. And the other one was Thickvein. So within 1 year, we had cloned Pond and Thickvein, published these 2 Cell papers. It was very exciting. Things went very fast. So Pond is a type 2 receptor. It's a serine threonine kinase, has a serine threonine kinase. That's what Connie cloned, based on the Thickvein receptor. Thickveins turned out, based on the TGF-beta receptor, sorry. Thickvein also turned out to have a serine threonine kinase domain, of course. And it also had a lot of serine residues, or a GS domain, that we will see what happens. Pond, type 2 receptor, Thickvein, type 1 receptor. These are the ones that bind the ligand from the 2 sides. I'll have to speed up a bit, but you'll get a 15-minute break as soon as we're done with this, before we start with the lab. Something that is important for today, for the lab, is a mutant that we call Thickvein QD. So for Pond and Thickvein, what we identified early on were, of course, loss of function mutations. That's what most of Jannie's, I think all of Jannie Nusslein's strains, these embryonic lethal mutations that she screened for, were loss of function alleles. So Pond, loss of function allele, means you don't have the functional receptor, so you don't have DPP signaling. Same for Thickvein. But for many things, people want to artificially activate receptors, or kinases in particular. People in that field early on, they realized that phosphorylation can be mimicked by adding aspartate, for example, amino acids that are negatively charged to a certain place, and that mimics the phosphorylation state of proteins. So people sort of learned that you can artificially, by changing amino acids, activate a protein by telling the protein you're phosphorylated in this particular place, by mimicking this phosphorylation. So people try to put in aspartate in all kinds of positions in these receptors to activate these TBP or BMP or TGF-beta receptors. And in the mammalian field, they found that actually it's not a serine that you have to mutate to an aspartate, but they made random mutations around the serines, of course. But they realized it's actually a Q residue, glutamine, I guess, to aspartate or glutamic acid. I'm not good in the peptide, in the amino acid business. But they found that they can mutate the Q to a D at a certain position, and this activates the type 1 receptor. The type 1 receptor is the one that activates the SMAD. So once you have an activated type 1 receptors, you activate SMAD and DPP signaling. So once you introduce this mutation into a receptor, the receptor doesn't need to see the ligand anymore. It is activated because of this mutation, and then you can activate the TGF-beta pathway. And this was very important to clearly show that DPP signaling is important for X, Y, Z, because you could activate the pathway sort of at the downstream level. And we'll use this mutation in the course. So Thickvein QD is, if you put this mutation in one of the chromosomes, the DPP pathway will be activated in every cell. So of course you cannot do it in flies, because you will not get an embryo once you start to activate all DPP signalings. You have to do that, let's say, using the DAO4 system, and we'll see how this is used. But this is a ligand-independent activated receptor. And if you want to show that gene A is activated by DBP, you want to show that it's also activated by this receptor, because then you have two independent evidences for this. So this is how one thinks these receptors are activated. The ligand comes, brings these two receptors together, type 1 and type 2, and then the type 2 phosphorylates type 1. This is then activated and phosphorylates MADS. And there are different receptor combinations that can be used, but for us, it's DBP, ThicVen, and POND. Again, maybe some structures. This is TGF-beta 1 is in light green, the two monomers here. This is the ligand. This is what I showed, this thing. And when you look at where the type 2 and type 1 receptors bind, the blue code here, activin receptors. So you can see that these are type 1 and type 2 receptors. Let's say this is type 2 and this is type 1. So the molecule is completely sort of embedded within these receptor complexes. So the receptor binds all this surface here and all this surface over here. And we have seen before that knock-in actually interacts here and over here with the receptor. So if knock-in is bound, the receptors cannot bind. If the molecule is bound to receptors, knock-in cannot bind anymore. And there is a large surface where these molecules bind with their receptors. It's a very stable interaction. The off rate is relatively low. So once a receptor has bound the ligand and has recruited the second receptor, then the molecule will not be released anymore. So the off rate, I think these have been measured there. The on rate is slow, but the off rate, also the on rate is kind of fast. And once the receptors are there, the off rate is there's no off rate anymore. The molecule stays there and then is endocytosed and degraded. And these again, with the structures you can see, that's a very stable surface. And so let's say this molecule would release here, this would still hold it. It's kind of, you're trapped. You cannot go out of this trapping anymore. This we can maybe loop it, but then this is what we still do not know. Structures are nice. We know what DBP looks like. We know what the receptor complexes look like. But how does that play out on the surface of a cell? Are there type 1 receptors that swim around interacting with each other and type 2 receptors swimming around, and then the ligand first brings the type 2 receptors in close proximity and then disassociate with the type 1 to make this complex? Or are there preformed complexes that just bind the ligand? Are there preformed dimers that then make heterotetramers? This is not well known. These are very simple questions, actually, kind of, because we have 1, 2, 3 molecules, some as dimers, so you have maybe 2 ligands, 4 receptors, 6 molecules. It's not so complicated. The complicated thing is that these are transmembrane proteins. There's no structure of a DBP or a TGF-beta receptor embedded within the membrane with its extracellular ligand binding domain and the intracellular serine threonine kinase. There's no such structure. And there is no structure of a full signaling complex here, of course, because you need the membrane, you need all of these, you need to, and then even if you have this structure, you still don't know how it comes about. How do you bring these components together? Having this structure doesn't show you the dynamics of bringing everything together. And at the end of the day, this is the key. As we said, molecules, they swim around. But swimming around TGF-beta type 1 and type 2 receptors, they don't do anything. They don't activate the pathway. And they have to be kept away from each other, because if you artificially link the type 1 and 2 receptors, they immediately activate the signaling pathway. We've done that early on already. So it's all about bringing these two together in cells that have seen enough of the ligand. And then the thing goes. And the whole dynamics here, that's biophysics, structural biology, molecular dynamics. Very interesting, because if this wouldn't work, this wouldn't work either. The system would either always be on or off. It's all about molecular interactions dynamics to make from this state to this state. Not solved. We do not understand biology at that level. Okay, the last molecule we look at, now we have the ligand that triggers receptor accumulation in certain places. Are these large clusters, maybe superclusters, or are they each individual receptor heterodimer is enough to signal? We also don't know. Where is this threshold for signaling? One receptor might not be enough. How many do you need? You might do different things at different levels of activation as well. But the logic is in these molecules. And here we come to the last molecule, the substrates. So the signal is now gone across the membrane because the receptors are together. And now if you look at the membrane from inside, you see these receptors seem to be clustered. Type 2 phosphorylates type 1. Now the serine threonine kinase of type 1 is also active. What does it do? How many molecules does the type 1 receptor phosphorylate? I only know, honestly, in flies of one molecule that is capable to be phosphorylated by the type 1 receptor, and this is SMAD. These are the SMAD proteins. This is the only one. I don't know about any other protein. I think PONT, the type 2 receptor, it's an active serine threonine kinase receptor. It doesn't need to be activated. It needs to come close to the type 1 receptor. But for thick vein in flies, the only molecule I know of that it can phosphorylate is SMAD. So the SMAD proteins, that's what they look like. They have a length of 455 amino acids. So they're encoded as 455 amino acids. No cleavage, it's a cytoplasmic protein. No single peptide, since it's a cytoplasmic protein. All 450 encoded amino acids fold into a certain structure. This structure has two folds in it, MH1, two homologies, which in structural biology turned out to be two folds, the MH1 domain and the MH2 domain. The MH2 domain of the SMAD protein has a SSXS sequence at the very C terminus. And since PONT is a serine threonine kinase, and this sticks out of the structure at one end of the peptide, this was a good candidate to be phosphorylated, and indeed these two serines are phosphorylated by the type 1 receptor. And this brings now the signal from a membrane-bound activated serine threonine kinase to a cytoplasmic protein, which now suddenly is phosphorylated. It wasn't phosphorylated before. And upon phosphorylation, it oligomerizes, and it oligomerizes with Medea and goes to the nucleus. So this phosphorylation leads to the nuclear translocation. It leads to the association with other factors. It leads in the nucleus to DNA binding. The MH1 domain is a DNA binding domain. And MH2 domain, in addition, is required for heterooligomerization with MAD and with Medea, the co-SMAD, and leads to transcriptional activation. So this molecule is inert in the cytoplasm. The phosphorylation of these two residues needs to oligomerization, nuclear translocation, DNA binding, and transcriptional activation. Two phosphate residues. You activate to this molecule, and there you go. It's a different world, completely different world. And that's, again, I like that very much. There is minimal things you have to do, and then boom, everything changes. Transcription changes then afterwards, and the cell is a different cell. It does different things now. This is what MAD looks like. The receptor SMAD in flies, this MH1 domain that binds DNA, the MH2 domain that interacts with the receptor. The receptor phosphorylates this. Once this is phosphorylated, the ligand is released. It binds with the MH2 domain to the co-SMAD. The co-SMAD cannot be phosphorylated. It doesn't bind to the receptor. So it's a kind of funny. Why would you need this heterodimer? I don't know. Nobody knows. Could have been a trimer of this going to the nucleus, but apparently it needs to be a heteromeric trimer, probably one molecule of this and two molecules of this, or then it's a hexamer. I don't think the structures have been solved for these, either trimer or certainly not hexamer. So these two molecules associate, they go into the nucleus. They're anti-SMADs. So if you want to design a molecule that is very powerful, you say, okay, I remove the 2-phosphorylation side of this molecule. So then it binds to the receptor, but it cannot be phosphorylated, gets stuck. It's never released anymore, and the pathway is shut down. And this is a bit what the SMADs look like. They can't be phosphorylated. They also don't bind to DNA, but they can kind of be recruited as Medea-like molecules, and then they go to the nucleus, but they can't bind DNA, and that poisons the system. Poisons the system by binding to the receptors, not being able to be phosphorylated. Poisons the system in the cytoplasm because it can be recruited to the SMAD, but doesn't have a DNA binding domain. These SMAD proteins, we talk about it, that these are encoded by genes that are first responders to the TGF pathway in every cell. Every cell, one of the target genes is that. That is a marker for which cells have seen DBP signaling. And it is believed that this downregulates the signaling response after it has come to a cell. So the cell responds, but in its response, it says, okay, I've seen it, I've heard it, leave me alone, expresses that and stops the pathway. Interestingly enough, that mutants are homozygous viable. So shutting down the pathway seems to be very important because all organisms have DAD proteins, SMAD, vertebrates. They're all regulated directly by the pathway, but in flies, the gene is not important. Kind of puzzling. We don't know why. Okay, we've seen this. We've seen this. No DNA binding, that's the logic. These are some of the structures of SMADs. Usually, alpha helices bind into major grooves in DNA binding proteins. Here, it's sort of a beta hairpin that recognizes the DNA sequences. So SMAD proteins recognize GC-rich sequences in the major groove. So GGC usually, and then since they are trimers or hexamers, these are sort of 3 GGCs, and we'll see that a bit in the afternoon. So here we go. We have started with a ligand, leads to oligomerization of receptors, heterotransphosphorylation, and then phosphorylation of a cytoplasmic protein, association of this cytoplasmic protein with another cytoplasmic protein, all of them DNA binding domains, have a large DNA binding complex here with 6 DNA binding domains, or 3 if they're trimers, and then it binds to DNA and regulates genes. That is the logic of the pathway. So here we go. We finish the first lecture. That's good. The questions that we ask now, this is the pathway. If I ask you now, how do I regulate genes differently by this pathway in the liver versus blood cells or brain cells? I mean, the liver will turn on SMAD or MAD, the DAD gene, that's this negative regulator. All of these cells will turn on that. But all of these cells will turn on genes that are specific for their own tissue, liver cells, blood cells, brain cells, muscle cells. BMPs do, or DBP does something else in each and every of these cell types. And that's what we want to understand. That's what we had no clue about in 93, 94, 95. The receptor was published 95, the second receptor 96, and then 2000, 2010, and even now, we still work on this. We figured out how all of that works, and you will figure that out too in the next 1.5 hours or so. So the questions that we are going to look at in the lab are is the signal transduced? How does the signal go into the nucleus? You know now some of the components, but now how are genes turned on or off in a tissue-specific manner? So now we make 15-minute break, which is until 10:35. And then I'd like you to come back, and I would even like you to come back with your screen turned on. As I can see now, there is only Kasha and David Ineichen with a with a fixed picture. None of the other, of course, has its glints. Now some turn up. So we make a 15-minute break, and then please come back, and it would be nice if some of you turn on the camera. So then it's a bit more interactive. I look very much forward to it. It's the first time you come with me to the microscope, to the pictures I make. Let's see how that goes. So we see each other again in 15 minutes.